Hi, so now I can be heard, I'm sure. No, it's no problem at all. I'm glad that we are together. There we go. Um, Laurence, do you go by Laurence? I can.
I need to unmute myself. I do go by Laurence, yeah.
Thank you. A bewildering array of choices, all for 99 cents or less. Or is everything actually 99 cents? It looks like you buy, if it's less than 99 cents, they get you to buy 2 or 3 of them. I think every purchase seems to be in a unit of 99 cents in this store.
I think so, yeah. I think I doubt these stores really exist anymore because of inflation. But there used to be Poundland, I think, which was the equivalent.
I'll probably unmute in 3 minutes or so, 3 or 4 minutes, and then we'll get started.
Sounds good.
All right, it's my pleasure to introduce today's Swartz seminar speaker, Professor Laurence Hunt from the University of Oxford in the UK, remotely by Zoom, of course. I see people are trickling in, but I think we have a quorum. So yes, Laurence got his training in Cambridge, in the University of Cambridge in England, in medicine, before going on to his PhD work in cognitive neuroscience at Oxford University, where he was co-mentored by Tim Behrens and Matthew Rushworth. And during this time, he ambitiously, I would say, combined different approaches, MEG, in particular, MEG and circuit models. And this became a sort of a theme that I think we can see through the different stages of Laurence's work, focused on questions about the formation of value representations. In 2012, he graduated and went on to his postdoctoral work, where he was a Wellcome Trust Fellow at the University College London. And in that period of time, he was further combining different approaches with Steve Kennerly and Ray Dolan as co-mentors again, where Steve provided essentially the perspective from the non-human primate and electrophysiology and recordings of neurons, and Ray Dolan, obviously on the human side with the use of fMRI, something that was Laurence had already done before while working with Tim Barons, but focused on more. And most recently, he's started his own independent lab back at the University of Oxford under the Henry Dale Fellowship, also supported by the Wellcome Trust. So it's a real pleasure to host Professor Hunt today. One of the types of questions near and dear to my heart is this effort to understand animal models and recordings in humans. And so I've greatly enjoyed seeing in the work that Laurence has presented, published, this sort of challenge being embraced and connecting MEG, fMRI, single neuron electrophysiology across animals and humans to study the dynamics of decision making and how it unfolds across different brain regions and how these networks are organized and support these computations, I think is obviously one of the great questions of our field. So it's with tremendous enthusiasm that yeah, you can't see me because I'm not allowed to be seen, I don't think. Yeah, I can. Oh, I can be seen. I apologize. My apologies. It wasn't working earlier, but now it is. Thank you, Xiaojing, for pointing that out. So yes, without further ado, I would like to introduce Professor Laurence Hunt to speak on frontal circuit specializations for decision making. Thank you very much.
That's great. Thanks a lot, Bijan. That's a very kind introduction. Just to check, you can see and hear me okay? Just get a thumbs up either in the chat or manually. Yeah, good. Okay. So yes, thanks a lot for the kind invitation, and I'm very sorry that it's not possible for me to come across the pond at the moment and visit in person, but such are these times. But in some ways, it is a very nice time to give this talk, as it's going to cover a lot of the same ground as this review article that I wrote just this past year for EJN. And so please feel free to take a look at this if you want to pick up on any of the references or findings that are going to be covered in this talk. So my talk today is going to look at how circuits within the prefrontal cortex support reward-guided decision-making in primates. And so I always think it's nice to start by showing you some primates making reward-guided choices in the wild. This is, of course, a very famous photograph by the German photographer Andreas Gursky. And I think that kind of one of the many reasons why this picture has become so famous is it kind of sets into contrast the almost bewildering array of choices that we're faced with, even on an everyday visit to the supermarket, versus the almost mundane nature of going about solving these choices. This is not a difficult decision problem for these decision makers to solve. And the reason why we can make decisions that appear as seemingly complicated as this one is, of course, that we don't consider and feed all of these different options into a comparison process and trying to maximize and find the best option that's out there. Instead, we treat this decision problem as a sequential problem where we consider one or a few options at a time by sequentially deploying our attention to different alternatives that are available. And so what I want to emphasize today is that process of decision making really being a temporally extended and a sequential and attention guided process is true not only of a lot of the kind of day-to-day decisions that we are faced with in our daily lives, but they'll also be true of a lot of the decisions that are faced in our kind of evolutionary ancestry, and so they will have played an important role in shaping the neural circuits within the frontal cortex, for example, that support reward-based decisions. So we can start by watching one of these decisions play out in the wild. So you can see here that our decision makers are faced with two alternatives, as is often the case in our lab experiments on decision making. But he's sequentially deliberating by looking backwards and forwards and considering their advantages and disadvantages before choosing what to go for. And there might also be examples like this one, where despite having lots of different alternatives on offer, he seems to be satisficing in some ways. He seems to just consider one of the options that's being presented and decides that it's sufficiently good, that it's past some kind of threshold, that he doesn't really need to deliberate and consider any of the other alternatives that are out there. And he can just go ahead and choose the option that he wants to go for. So this kind of immediately gives rise to questions about how attention and choice are interacting as we go about making value-based decisions. How do we decide whereabouts we're going to deploy our attention next? How do these shifts in gaze that we are making actually shape the decision that we come to in the end? How do we decide when we've sampled enough information that's out there and when to stop and choose an option and then move on to the next decision? And then, importantly for this talk, what are the neural representations within prefrontal cortex that support all of these different processes, and how might they differ across different prefrontal subregions? So this idea of attention being an important factor to consider in value-based decision-making has now received a lot of attention in the behavioral economics literature. And here's a very influential early study from Ian Krybik and Antonio Rangel that looked at this, where they asked hungry human subjects to deliberate between different snack food items that they might want to eat at the end of the decision, but crucially, they allow them to freely move their eyes as they're making their decision. And what they find is that their participants' choices and their reaction times are very well described by a drift diffusion model, a common model of evidence accumulation in the neuroscience and cognitive psychology of decision making. But crucially, this evidence accumulation model has a key twist, which is that the speed of evidence accumulation is going to depend upon where the subject is currently attending to during the decision process. So you tend to accumulate more evidence for the item that you're currently looking at. And as I'm sure many of you in the audience will be aware, this feature of the model then allows the authors to capture several key choice biases that emerge in the behavioral data that you wouldn't be able to get if you just considered it as a standard drift diffusion model. So from this work, we kind of know that the locus of visual attention seems to be having a strong influence on how values of different items are going to be weighed as a decision unfolds over time. And we also know that in certain circumstances, this influence can go in the opposite direction, in that attention can be very rapidly oriented towards objects in the environment that have high value. So here's an example of this from a study by Sean Kavanagh, who was a PhD student with Steve Kennelly and myself, looking at a very similar decision task in macaque monkeys, where they are presented with two pictures telling the monkey about the reward magnitude or probability that's on offer. And similarly to the Krybik study, they're making a choice with a joystick, but they can look at the pictures with their eyes as they're making that decision. And what we found to be fairly remarkable about this data is that even though the joystick choice tends to take about half a second on average, the monkeys make a first saccade within 150 milliseconds or so to one of the pictures, and that very first saccade tends to be driven towards the picture which is more valuable. And this is particularly true for pictures which the monkey has got a lot of experience with, the ones which we're here labeling as overtrained pictures. So from this, it kind of appears that there might be an interaction between attention and choice, which is like a two-way street. So your attention can be captured by items in the environment, which are likely to be more valuable. And this in turn can also influence the likelihood of you going on to choose that item in the future if you spend more time looking at it. Now, of course, the two alternative forced choice paradigms that we study in the lab are really quite artificial in nature, but I think that they emphasize that we need to start to think about decision making from this more sequential perspective. And if we observe how primates make decisions in naturalistic environments, I would argue that this also kind of points towards these are the kinds of decisions that we may have been evolved to make. So here are two monkeys that are foraging for foods in very different environments, but you can see that some features of their behavior are common. They're relying very heavily upon attention as they saccading around the environment as they search for food, and they perform this search in a sequential fashion. And in their book on the evolution of prefrontal cortex, what Dick Passingham and Steve Wise argued is that primates forage in a very distinct evolutionary niche from other mammals. Because they are diurnal rather than nocturnal, their foraging will rely more upon visual attention. They have forward-facing eyes and a fovea, which will allow very fine visual discrimination of objects in the distance, and that's particularly important in this fine branch niche where they have to coordinate where those objects are in space with reaching dexterously for those items in order to collect them. And they can also learn extremely rapidly based upon those visual cues as to locations that are likely to lead to reward and those which are not. Often they rely upon individual visual cues in order to shape their behavior about where to forage next in the environment. And so what Passingham and Wise argued is that this is the reason why, at the same time as we saw in a primate brain, this dramatic expansion of the visual system and the development of these fine hand movements, primates also showed this dramatic expansion of the prefrontal cortex. And in particular, they developed the granular prefrontal cortex with an expanded layer 4. And of course, many of these regions then go on to further expand and differentiate in the human brain, but they're largely absent from a rodent. And so I think that if we are going to try to understand how different subregions within the prefrontal cortex support value-based decision-making, it makes sense not only to study it in the primate brain, but it also makes sense to try to understand the role that visual attention might play in shaping our choices. And so what I want to emphasize in today's talk are two ways in which the circuitry within the primate prefrontal cortex might be specialized for performing sequential temporally extended choices. So in the first half of the talk, which will be rather shorter, I'm just going to spend a few slides talking about the microcircuitry within prefrontal cortex and what this predicts about the dynamics of value correlates during temporally extended tasks, and how recurrent microcircuits give rise to particular sequences of value correlates when making simple value-based decisions. And then in the second part of the talk, I'm going to be discussing the macrocircuit connectivity of different PFC subregions, how PFC subregions differ from one another in terms of where they talk to, and how this might shape the neural representations that we see within those subregions during temporally extended and attention-guided choice. Okay, so let's first of all think about the microcircuitry. So here are some illustrations of layer 3 pyramidal neurons from monkey primary visual cortex on the top row and lateral prefrontal cortex on the bottom row. And what you can see morphologically is there's just a very striking difference in terms of the sheer size of the dendritic tree of these prefrontal neurons. And what's more, there's also a greater density of the large dendritic spines along the dendrites within PFC neurons than there is in early visual cortex. Now, because these dendrites are going to primarily be receiving cortical projections from other nearby neurons, this means that prefrontal cortex will have a much greater degree of local recurrent excitatory connectivity than early sensory regions. In other words, a neuron in PFC typically receives about 5 to 10 times the number of local excitatory connections from other neighboring neurons relative to a V1 neuron. And just for comparison, this is this difference between neuronal morphology in V1 versus frontal cortex really doesn't seem to be present to the same extent in the rodent brain. And this is from a comparative study from Jennifer Lubke's lab, which also contains a lot of the morphological and physiological differences between these different regions across species. So this is a point which many of you at NYU will need no introduction to, because it's the basis of a lot of work that's emerged from Zhao Jing's lab over the past 10 or 15 years or so now. And what you can see is that difference in dendritic spine counts that I showed you on the previous slide is actually part of a more gradual variation that's seen across the cortical hierarchy. And this in general correlates with the hierarchical position of an anatomical area defined in terms of that region's connectivity to other brain regions. So in other words, those regions that we think of as being closer to sensory input and lower within the anatomical hierarchy will have neurons with a lower dendritic spine count, whereas those at the top of the hierarchy, which are receiving more highly processed sensory information, will have many more dendritic spines and much more in the way of local recurrent excitation. And what Zhao Jing and John Murray beautifully showed is what this translates into physiologically is that activity within the higher cortical areas tends to be more persistent over time, even when the animal is at rest. So this was shown by examining the spike count autocorrelation of neurons across these different cortical regions. And what's found is that the autocorrelation decays far more slowly in these areas which are higher in the hierarchy, which have more in the way of local recurrent excitation. And I'm sure many of you are aware that this has been further built upon by work by Zhao Jing and John and others, showing that this is mirrored in many other circuit properties, such as NMDA receptor subunit composition, D1 receptor expression, and so on, and has started to be used as a basis for really beautiful large-scale computational modeling of the interactions between these different regions. So what we were interested in was how we might take these recurrent networks that we know exist within the prefrontal cortex, which are relied upon for solving temporally extended cognitive tasks, such as working memory or evidence integration or sequence production, and to ask, well, what can we use from these network models? What can we predict from them in terms of how a value-based decision might be made, and what might emerge from that network in terms of value correlates as a decision unfolds. And so this is work which is a few years old now, but so I just want to give the kind of high-level take-home messages from it. But what the takeaway from this work is that we performed this series of studies where we hit these networks with a large number of different decisions where we manipulate the value of the different options that are being presented to the different selective pools. And then we ask, well, what are the value correlates that then emerge across different trials within the single units and more macroscopically across the network as a whole that might mirror what we would see within the local field potential as well. And so we can make these predictions about a range of different types of value correlate that we might see relating to value difference and chosen value and the eventual choice that's being made. And the key prediction is the relative timing and the sequence of different value correlates and when they emerge relative to one another as the decision is unfolding. And so what we found is that there's a very good correspondence between what these local recurrent excitatory networks are predicting in terms of a sequence of value correlates and what we see in the neuronal data, both within the macaque single unit recordings and also using the LFP as a proxy for what we might see at the scalp using human MEG recording. And one point that I just want to kind of particularly emphasize when thinking about these dynamical value correlates within these network models is actually how they're arising within the model. So what I'm plotting here are the average responses of this network model, which has these winner-take-all dynamics. So it's approaching one of these two attractor states as a decision is being made. It's either choosing option A or option B on different trials. And we're sorting the responses of the network from trials which are very easy, where the chosen value is much higher than the unchosen value, or trials where it's a much more difficult decision for the network to make. So where the comparison being made is much closer. And the key thing that you can see is that the process that's unfolding within the network is exactly the same process on all of these different trials. It's still a winner-take-all process that's approaching one of these two attractor states. But the inputs to the network are determining the speed at which that dynamical process is going to unfold across the trial. And it's the fact that this progresses at different speeds on different trials that determines the fact that you see these value correlates across trials. And so rather than the value correlates being something that the network has to represent per se, or signal to another brain region, these are kind of emerging more or less as an epiphenomenon of a fact that you have a dynamical process which is unfolding at different speeds on different trials. And so when we have neurophysiological recording data from PFC, we wanted to ask, well, is there anything that we can see that might test whether this is really the case, that's why we see these value correlates during a decision within the frontal lobe. And there were a couple of key features that we see in the data that suggest that it might. So when we look at the local field potential recordings within PFC, during this kind of simple two alternative force choice task that we face the monkeys with, we find that the evoked potential across a range of different, a few different subregions looks like this. So we see a standard evoked potential, but then at the bottom I'm plotting what when we see the value correlates for the chosen and unchosen value across time. And the key thing that you can see is that the time when the value correlates are maximal isn't the time when the LFP is peaking, it's the time when the LFP is ramping, it's when it's changing most across time. And so it's the speed at which this ramping process is changing over time that's giving rise to these value correlates emerging across trials. So it's that this ramp happens faster on trials where it's an easier decision for the network to resolve. And I don't have time to show you this, but within this paper, we also show that this ramping process in the LFP also explains the emergence of value correlates that we see in the single unit data that we're recording simultaneously as well. Okay, so that's a kind of a brief taste of some older work, which was looking at local recurrent microcircuitry and why that might give rise to certain value correlates and sequences of value correlates within PFC. And then for the remainder of my talk, what I want to talk about is, well, how different PFC subregions differ from one another in terms of their macrocircuit connectivity to other PFC subregions, and how this might shape the representations that we find when primates are performing temporally extended and attention-guided choice. So you can see on the left that prefrontal cortex, as I'm sure you're all aware, can be divided into a very large number of psychoarchitectonic subregions. And these regions, although they have strong connectivity to one another, they differ in terms of their connectivity to other cortical areas. And this is a point that's really beautifully emphasized in this review by Dick Passingham, which is a couple of decades old now, where he developed this way of plotting the connections of each subregion to other subregions, which he calls a connectivity fingerprint, where you can kind of quickly see the similarities and differences between the afferents and efferents of each subregion. So that was originally developed based on tracer data, which is of course very laborious to collect for all of these different subregions. With modern imaging techniques such as diffusion MRI and resting state fMRI, it's also now been possible to build up connectivity fingerprints for all of these different subregions within individual animals, and to do so across species. So here is a study from Matthew Rushworth's group in Oxford that does exactly that, focusing on different areas of PFC that are known to be involved in reward-guided decision-making and learning. So here, for example, is the connectivity fingerprint for area 13M in orbitofrontal cortex, showing data from the human brain in red and the monkey brain in blue. And what you can see is that there are very strong connections from 13M to regions such as the temporal pole and perirhinal cortex that's going to contain very highly processed sensory information right at the end of a ventral visual stream. So this will mean, of course, that the orbitofrontal cortex will receive information about whatever object the primate is currently attending to in space. By contrast, if we look in areas such as 24C in anterior cingulate cortex, these don't have such strong direct projections from sensory areas, but they do have strong connectivity to more motoric regions, such as primary motor cortex and supplementary motor area. And so ACC might be particularly well positioned to guide selection of actions as a decision is being made. And then in a prior study, this same group also examined the connectivity fingerprint for areas out on the lateral surface. And here's one such region, ventral area 946 in the dorsolateral prefrontal cortex. And this has very strong connections to parts of the inferior parietal lobule, which are parts of the dorsal visual stream, which we would closely associate with reorienting of visual attention. So it's with these kind of differences in connectivity between these different PFC subregions in mind that I want to tell you today about the recordings that we made from macaque monkeys as they performed a kind of temporally extended value-based decision-making task, which was guided by visual attention, where we positioned our recording chambers so that we were able to record populations of neurons, about 10 to 40 neurons a day, from each of these three different subregions. And many of the analyses that I'm going to show to you today are really relying upon the fact that we do have a reasonable number of neurons that we're able to perform population analyses of these data. But I should emphasize that everything that I'm going to present is based upon pseudo populations. And so I think that there's a huge amount to be gained in the future once we're able to record really large populations simultaneously from these different subregions as well. Okay, so whilst we recorded from these areas, we were getting our subjects to perform this temporally extended decision task guided by visual attention. And the task works like this. Subjects have to choose between these two options presented on the left and the right-hand side of the screen, each of which is consisted of two pictures. And the pictures are going to inform the subject about the reward probability and the reward magnitude that's associated with that option. And the subject has already learnt the meaning of these pictures extremely well before the recording session begins, but they're going to just be randomly sampled from these different pictures on every individual trial. So just to make this clear, so on this trial, the subject's choosing between a 70% chance of getting 1 drop of juice if you were to choose the left option versus a 10% chance of getting 5 drops of juice if you were to choose the right option. But the key trick in this experiment is that we are not going to show all of this information to the subject at once. Instead, we're going to force the animal to reveal the information sequentially by making saccades around the screen in order to gather information as the decision unfolds. So the first thing that appears is this fixation point, and then the subject sees the different locations, one of which is going to be highlighted, instructing the subject to make a saccade towards that location and revealing the picture hidden beneath. He then fixates that cue for 300 milliseconds, and then another picture is going to be highlighted. On half of the trials, that picture will be on the same side as the first one, and so then he will know both the probability and the magnitude associated with one of the options, but not know anything about the other option. So we can call those option trials. On the other half of the trials, the highlighted location will be on the other side of the screen. And so the subject then has learnt both pieces of information about one attribute, but doesn't yet know anything about the other attribute associated with those options. So we can refer to those as attribute trials. And then once both of those pictures have been sampled, the subject essentially becomes free to do as he chooses. So he can either go ahead and gather the remaining information and make saccades towards those locations, or he can terminate the trial at any stage by moving the joystick that he has in his hand to decide just based upon the information that's been gathered thus far. And then once the choice has been made, then all of the pictures are going to be revealed, and after a short delay, the juice reward will be delivered probabilistically with the magnitude of the chosen option. Okay, so that's how the task works and how we allow the subject to have this kind of interaction between where they're attending, leading up to the final decision that they're having to make at the end of the decision process. I'm not going to spend a huge amount of time on the behavior in this task because I want to get onto the neural data, but I'll quickly mention a couple of things. So first of all, the subjects know the values of these cues very well, and they use this appropriately to their advantage. So they end up choosing the best option on about 80% of the trials in terms of normatively choosing the option with a higher expected value. But they can do this even without gathering all of the information before committing to their choice. So they don't tend to gather all of that information. In fact, on only about 15% of the time do they invest the time needed to go and look at all of the 4 cues. The remainder of the trials, they go ahead and gather and make their choice based upon 2 or 3 cues. Oh, sorry, my animations have gone. All right, there we go. And so that raises the question about, well, how do they choose to gather this information? And there were a couple of pieces of evidence about this in the paper, but the key takeaway from that analysis is that subjects tend to want to gather more information about the option that they currently prefer. So they don't view information against an option as being equivalent to information for an option, if you see what I mean. So the subjects want to gather more information in confirmation of choosing the option that they end up wanting to select in a trial. And I can show the evidence for that at the end of the talk if people are interested. Okay, so that's the task. So how do we then go and look at the neuronal data from our different prefrontal subregions? Well, the one approach that we found to be particularly informative in terms of distinguishing the different representations that we found within prefrontal cortex during this task was to use representational similarity analysis. And I'm sure this is familiar to most of you, but I'll just kind of quickly walk you through the approach. So what we do is we look at the responses to a particular cue in a particular location, and we will plot the firing rates along the x-axis in response to viewing that cue. We've z-scored them here. And then we can take that same neural population and ask, well, how do those neurons respond on a different set of trials where the same cue is being presented over on the other side of the screen? And if we see there's a positive correlation, it means that there's some population similarity between those 2 conditions in terms of how the brain region responds. And so we can go ahead and repeat that process for different combinations of cues, and we might see a negative relationship or positive relationship in different conditions. And we can use this to build up a matrix of correlation coefficients that's going to tell us how similar or dissimilar each of these 10 cues are to one another when they're either presented on the left option or when they're presented on the right hand option. And so if we look at this for OFC, to begin with, when we did this, we can see that there's this kind of really beautiful structure which emerges in this RSA matrix of how similar the responses are to these different conditions. So just to kind of orient you to how this matrix is being sorted, we've got the probability and magnitude cues sorted from the worst to the best, from lowest ranked to highest ranked here. And we've got them when they're presented on the left and right-hand side of the screen. And you can hopefully just kind of see by visual inspection that there are things which just pop out of this matrix as being potentially interesting. So first of all, we have these bright off-diagonal elements here, which indicate that the OFC responds similarly to the same picture irrespective of whether it's being attended to on the left-hand side or the right-hand side of the screen. So OFC seems to be encoding the identity of the currently attended picture. And we also have these squares that are kind of repeating themselves, 5 by 5 squares across the entire matrix. And what this reflects is a value response for the currently attended value. So in other words, low value pictures are going to be positively correlated with other low value pictures, and high value pictures are positively correlated with other high value pictures, but low and high are going to be more anti-correlated with one another. Okay, so that's what it looks like for orbitofrontal cortex. But what about the other two regions that we're recording from in this task? So if we repeat the same analysis for both the ACC and the lateral prefrontal cortex, we see that there's some similarities between these different regions, but there are also some key things which are really quite different between these three different subregions. So first of all, neither ACC or DLPFC seem to carry any of the information about the current stimulus that the subject is currently attending to. Instead, in ACC, we do see something that looks like the value code that I just highlighted in OFC. But interestingly, it seems to take on slightly more of a block-like structure, almost as if it's kind of categorizing the cue into, is this an item which is above average in value? Is it one that I would want to choose? Or is it below average? Is it one that I might want to reject? And so we'll come back to this idea of committing to a decision in ACC a little bit later. And then lastly, in dorsolateral prefrontal cortex, we see very strong spatially lateralized responses. So LPFC really strongly encodes the currently attended spatial location. So this was all time locked to just after the first cue was presented, but we can also, of course, we can repeat this process and just slide it across time to kind of ask, well, when do each of these different representations emerge relative to one another? And to get a little bit more formal about some of these different representations, we build up templates that capture these different features, and then we regress them onto the RSA matrix across time. And so what you can see now is the change in the RSA matrix across time. The subject's just saccaded towards the stimulus, and you see encoding of that spatial attention template emerges early on in DLPFC. So it seems to be more time locked to when the saccade is occurring. And then the other representations relating to attended value and accept reject kind of emerge more or less simultaneously across OFC and ACC. And then at the very end, we see some lateralized value signals, which might code value specifically for whether I'm going to make a leftward or a rightward action, which is, of course, the key thing which is relevant for making the final decision subsequently after more cues will be collected. Just as a kind of a little bit of an aside from this monkey study, RSA has also been popularized as an approach to try to bridge between what we can see in non-human primates with what we can see in humans. And so we've recently tried to do the same, use the same paradigm in human fMRI data. And this was a project led by my graduate student, Paula Kanders. And the results here surprised us a little in that although we can find reliable representations of many task features using RSA on the fMRI data, we don't seem to see a very clear correspondence between the anatomical dissociation that we can see in the monkey single unit recordings and what we can see in the human prefrontal cortex. And in some ways, you know, this may be disappointing in that, you know, RSA has been touted as a way to potentially bridge across representations that we can see across species. But I also think that it may point to the fact that, you know, fMRI is by its very nature going to be averaging across very large numbers of these neighboring neurons. And so unlike in regions such as the ventral visual stream, where we have columnar organization, and so multivariate patterns may still be sensitive to picking up on things which are common between humans and primates. We can't really use RSA in quite the same way as a bridge across species in prefrontal cortex. And this actually matches with very nicely with a paper from Doris Zhou's lab back in 2015, asking about where the limits lie in applying RSA as a bridge between what you can see in equivalent fMRI data and single unit recordings from the same brain region. Okay, so let's now return to the monkey cells. So remember that all of the analysis that I've shown you so far is just when the very first picture has been attended to by the monkey, but in fact there are now opportunities to go and sample more pictures before committing to making a decision. So what happens as the monkey starts to accumulate more evidence before committing to making a choice? So let's consider this in the orbitofrontal cortex, where the monkey is shifting his gaze from one side of a screen over to the other side of a screen on the attribute trials. And we can see that there are these nice value correlates that emerge time-locked to each cue being fixated in the single unit data, but these don't return all the way back down to baseline. So it looks like there's some memory of the previously attended cues that persists in orbitofrontal cortex. So the first question that we want to ask is, well, does this value response that we see time locked to Q1 happen in the same set of neurons to the value response that we see when we're looking at Q2? So the way that we're going to do this is we're going to plot the regression coefficients for this attended signal when I'm looking at Q1. And what we find is that there's a very strong across neuron correlation. So each dot here is one neuron, and we're plotting how strongly you encode value of Q1 when you're looking at Q1 relative to how strongly you encode the value of Q2 when you're looking at Q2. In other words, this is a very stable population code that's reflecting the value of what's ever currently being fixated by the animal. But we can then go and play the same game where we are now comparing the population code whether these are the same neurons which are going to be encoding the memory trace of the previously attended cues as the monkey shifts his gaze. And what we find is that the population codes are now related, but crucially, they're negatively correlated with one another rather than positively correlated. So in other words, when I'm attending to the second cue, the neurons there's an inhibition process which seems to be taking place between the population code for the currently attended item relative to the items that were previously attended and stored in working memory. And that's just highlighted at 2 time points, but in the paper we can build this up into a matrix of these different correlation coefficients across time. So B just refers to when we see this very strong positive correlation, and C is when we see this negative correlation here. And this is useful because it gives us a very clear way to just compare what we see across the 3 different subregions. And so what you can see is that although DL, PFC, and ACC do carry a value code for whatever you're currently evaluating, it only seems to be OFC where we have evidence of this inhibition process between the currently attended and the previously attended items that have been being stored in working memory. Okay, so that's what's happening in OFC, but what about in ACC? Well, what we've been able to do here is that we find that there's a population of neurons that appear to increase their activity whenever the monkey sees a new piece of information that confirms their belief about which item they currently want to choose, whether they should pursue the item that they currently think is their current best option. So we define a bunch of regressors that we use to look for a code which seems to suggest that there's a belief confirmation code there in ACC. And what we've done here is we've then projected data from the ACC on a left out subset of trials into that subspace, and then asked, well, how does the activity evolve within that subspace on trials of different reaction times? So these are trials where the subject responds very early with the joystick, commits to making a decision early, whereas these are the ones where the subject takes a long time, samples a lot of information before they're prepared to commit to a final decision. So if we slide this across so that it's response locked, you can see that this peaks just a couple of 100 milliseconds prior to the time when the subject commits to making a final choice. So it seems like there's a kind of threshold that's reached within this belief confirmation subspace prior to the decision. And what's also interesting is that there's also neuronal activity in ACC that's not only selective for a decision that's going to be made, but also whether that's going to be a rightward movement or a leftward movement that's being committed to at the end of a decision. And so when we project our data into this separate orthogonal subspace, reflecting the representation of which action will be chosen by the subject, you see that both of those things are kind of emerging and ramping up simultaneously prior to the decision. Okay, so I'm almost out of time. I just want to quickly finish up with a couple of interesting follow-ups on ACC and the role that we think it may be playing in information sampling in this task. So the first is that there's actually a bit of a couple of Easter eggs in this data set, and one of which is that we actually have a third of the trials in every session, which we don't talk about in our 2018 paper, where the subjects, where all of the cues are appearing simultaneously, and the subject can just freely saccade and choose whenever they want to. And so James Butler in Steve Kennerly's lab has recently gone and analyzed this data, and going right back to the start of the talk when I was telling you about these kind of covert evaluation of stimuli that are in the periphery of the monkey's visual field. What he finds is that on these trials where this information is presented, when the animal makes the very first saccade towards a cue, we not only see encoding of the currently attended cue, but we also see in ACC weak encoding of all of the other alternatives that the monkey could have saccaded to. And what's particularly interesting is that trial by trial changes within how strongly those other items are being encoded is then going to predict where the animal is next going to make future saccades, about whether they're going to make vertical saccades within the same option or horizontal saccades over to the other side of the screen. So that's when everything is completely free for the animal to choose. And if you want to find out a little bit more about those analyses, that's papers up on bioRxiv. And then related to this, as I mentioned earlier on, is this fMRI version of a task that Paula ran in my lab. And the headline finding from this study, beyond the kind of null RSA results that I was talking about earlier, is that at the time when the second cue is presented, and we see something that looks kind of like a classic inverse value difference signal in the bold signal in the dorsal ACC. Subjects are being given this chance to decide to sample more information before making their choice. And crucially, when you include another regressor for information sampling in this regression model, what we find is that the parameter estimate for difficulty is reduced, and it actually becomes non-significant. And instead, the variance which we normally attribute to being a difficulty signal is actually better thought of as a decision as to whether or not to go and sample further information before committing to making a decision. So I think that this kind of opens the door maybe to start to think, well, how much of these ACC signals that we've studied really quite extensively in bold fMRI relating to value comparison might be thought of as the formation of a decision process versus the kind of internal decision to go and sample more information before committing to making a final choice. Okay, so with that, I'm going to close, and I just want to kind of quickly summarize the take-home messages from the second part of this talk. So we've seen that the three regions that we've recorded from in this study, due to the different anatomical connections that I highlighted, play quite dissociable roles in the task. Dorsolateral prefrontal cortex we saw had very strong representation of current deployment of spatial attention as the subject saccaded around the screen. OFC seemed to carry this population response that had this comparison process between items which are currently being attended versus those which are stored in working memory. And ACC seemed to have these population responses relating to whether the new information is sufficient to kind of confirm your current belief, and that ramped up prior to the commitment to making a final choice at the time when the decision was committed to by the subject. And so with that, I'd just like to close by kind of thanking the very many people who've been involved in this work across the years, but I'd particularly like to thank Nishmal Alasekara, who's joint first author on the 2018 work, who was a PhD student, very talented PhD student, who was working alongside me in Steve's lab on this data set. And I'd also like to thank Wellcome, NARSAD and Royal Society for their funding. So thanks very much.
Wonderful, thank you very much. So we actually have a question in the chat, or the Q&A, excuse me. And please type in your questions. I think my job obviously is to read them aloud and let the response. So we have a question from Sean Friedrich-Wolfs. A very interesting experiment. This is from the first half of your talk, I should say, the first part of your talk. Did you assess whether the monkeys were optimizing their reward rate over time? For example, by skipping items of information.
Yeah, so we definitely don't think that the reason why they're skipping over that information is for optimizing reward rate, although it would be interesting to do a kind of more formal analysis of that. I think that I guess the reason why we don't think that is that the relative amount of time that subjects would have to deploy to sample the additional pieces of information is of the order of 300 to 600 milliseconds or so, and the total trial length, once you've factored in all of the time for initial fixation and moving the joystick and so on, is of the order of about 20 seconds or so. So the relative time that would be needed to gain that additional information, I don't think it suggests that they're behaving optimally. I think it's more likely to be the case that they want to get to the reward sooner, and monkeys tend to be impulsive and will have strong discounting of early rewards. But it is something that would be an interesting thing to look at, the degree to which optimization of that does take place.
Fascinating, it makes a lot of sense. So now another question from Professor Chiani. Great talk. Is shape representation in OFC and the associated representation of value generalizable to other visual features such as motion? And there's a follow-up as well. I'll read it so you can decide. Is the function of OFC in your studies incidental to the design, which makes visual shape the most informative feature? So do you think it's generalizable, and how central to your
it's a good question. So I'm not sure. I mean, certainly the position of where OFC sits in terms of receiving highly processed sensory information from the top of the ventral visual stream kind of suggests that it would be sensitive to, and it also receives, polysensory information from other regions such as the auditory cortices and so on. I think would suggest that for many tasks that involve any form of translating sensory information into value, OFC would be a kind of natural node for that to take place. I was just trying to think off the top of my head if there were any examples of OFC recordings where it's a translation of motion into value that speak to that, but I can't think of any off the top of my head. So I guess the answer is that I would give is really that it's because of the connections to regions which are interested in visual shape, and because visual shape is very important for solving our task, that we see the representations that are associated with value coding there.
So I have a question. I'm always a broader question. I'm always interested when we see these, specializations across different brain regions that we know are heavily connected, and in particular over time. And you've touched on this, but I wanted to get your thoughts sort of whole in this question period in a sense. So when you see ACC neuron selectivity and information ramping up, and you know that those neurons, at least some large number of them, must project to, a decent number of them must project to OFC. Mm-hmm. How do you think the chain of causal sort of flow works, where these spikes are being generated? They're obviously significant, but OFC isn't really responding to them, or how would you express those thoughts about OFC's response to that anatomically defined input?
Yeah, so I mean, obviously, so let's say OFC is receiving information back from ACC that's coding like whether I'm integrating evidence towards a leftward action or a rightward action. I guess there would need to be a reason why that would be a useful thing for OFC to then make use of. I guess what I'm imagining would happen is that OFC would need to perform some kind of computation on those inputs for it to persist and be integrated within OFC and emerge as a representation there as well. So in other words, I could imagine a way in which you could send inputs back to OFC, but they're basically just being projected back into a null space within OFC spiking activity. And so so then the OFC representation is not going to show the even though it's still receiving inputs that are
yeah. So how do we get out of the null space, I guess, is where this that leaves us in OFC. So OFC's right.
So OFC doesn't want to get out of the null space, right? Because OFC is its job is to be listening to inputs related to what I'm currently attending to, and translate them into actions into a value code that then would be sent to ACC. ACC would want to translate that into a space which is encoding an action, and then that will be sent on to motoric structures. Now, I'm assuming that when it's going to motoric structures, it's not being projected into a null space, it's being projected into an action execution space. But yeah, it would be kind of like the transformation that needs to take place. You'd need to set up that, even though the same information is being sent to different regions, what's being done with that information should differ depending upon.
So we have these regional subspaces, which include, of course, null spaces, which are not responsive to within which they're not responsive to input, and that these regionally specific spaces are central to the computations, of course, the regions are performing, And that's how we should think about what's going on between the regions, at least in part.
That would be my yeah, but I mean, but I don't have strong evidence for that. I don't have population recordings of either of those, and I think it would be there's still kind of like an open question as to whether you can really build up a population model that does those transformations, where you have like reciprocal connections from OFC to ACC, where you are passing information that's being translated into a relevant code, being translated from a good space into an action space, but nevertheless, the reciprocal connection is not then re-influencing the activity back in OFC. Yeah, so I think that's a really interesting way forward for how to build a kind of multi-regional model of that, and then test that in simultaneous recordings.
No, fantastic. No, thanks for sharing that thought. We have any other? I think we've got a little bit more time. Are there any other questions in the audience? We'd be happy to entertain them. But I think we've learned a lot already from what you have taught us. So I think we can say thank you very much.
Great. Thank you very much for the opportunity to come present.